Now presenting our final presentation on cell adhesion.
All right, so this slide here is just an overview of cell adhesion. So basically, what is cell adhesion? Cell adhesion is the binding of a cell to another cell's extracellular matrix. Um, there are four families of cell adhesions. It's the immunoglobulin superfamily, cadherins, selectins, and integrins. Those are the four families. Um, the immunoglobulin superfamily. That is they use antibody proteins that attach to a foreign invader as part of the body's immune response. So selectins, they're plasma glycoproteins, and they bind to a specific carbohydrate that are located on the target cell. Cadherins, they're also a family of glycoproteins, and they mediate calcium-dependent adhesions between different cells. And integrins, they're combining of the cytoskeleton and they with the ECM proteins. And an example of that is probably collagen, something like that.
Immunoglobulin superfamily proteins use antibody proteins that attach to foreign invaders as part of body's immune response. They participate in heterophilic as well as homophilic adhesions. They contain loops that bind homophilically with adjacent cells. There are five major classes. The five classes are IgA, IgD, IgE, IgG, and IgM. The IgA class play a role in mucosal immunity. The IgD has antibody expressed in plasma membranes of immature B lymphocytes. The IgEs play a role in allergic disease. IgGs are protein complexes that contain four peptide chains, and IgMs contain the largest antibody and appear in response to antigens. All right, so selectins, they are a single chain transmembrane protein. There are three families involved, which are the P-selectins, E-selectins, and L-selectins. The P-selectins appear in platelets. The E-selectins are usually seen on endothelial leukocyte adhesion molecules, and they require transcription pathways which ultimately turn on endothelial cells. The L-selectin is a cell adhesion molecule and is usually found on lymphocytes, and the leukocytes invade the blood vessels. And here is a picture depicting that.
Okay, so cadherins are transmembrane proteins that rely on calcium ions to function. They play a role in cell adhesion, forming adherent junctions to bind cells within their tissues. They also help with tissue structure and cellular movement. They behave as both receptors and ligands for other molecules. The disruption of cadherins can lead to proliferation and uncontrolled cell migration of tumor cells during the growth of tumors. During tissue development, the tissue is dependent on the activities of the cadherin families, which are includes classical cadherins, which are those right here, the N, P, and E cadherins, protocadherins, and atypical cadherin. The extracellular domain of cadherins regulate homophilic and heterophilic interactions during adhesion and cell sorting. So there are three major types of cadherins. The N cadherin, which is found in neurons and found in neural cells. P cadherins found in the placenta, and E cadherins found in epithelial tissues. So E cadherin is a type 1 cadherin found at the adherent junctions. It is an adhesive receptor because it has a glue-like ability during the early stages of embryogenesis. They can be expressed in both polarized but as well as differentiated epithelial cells. Cells require E cadherins because, like in their presence, they remain tightly associated with the epithelium. But without the E cadherins being present, many of the cell adhesions as well as cell junction proteins in the epithelial cell will not be capable of supporting intercellular adhesion. Okay, so adherent junctions are protein complexes that occur at the cell-cell junctions in the epithelial and endothelial tissues. It can be defined as a cell whose cytoplasmic phase is linked with the actin skeleton. The adherent junctions is composed of a B cadene, a delta cadene, which is also the P120 cadene, and alpha cadene, and a gamma cadene as well. And so what cadenes are is that they're a family of scaffolding proteins. And a scaffolding protein is a regulator of many key signaling pathways. And their job pretty much is that they are supposed to interact and bind with multiple members of signaling pathways. And that is what catenins do, is that they bind with cadherins and they form complexes within these cell adhesion molecules. And so the primary role for a catenin is connecting the cadherin to the actin filament, which is specifically in the adhesion junction of the epithelial cells. Catenins play an important role in normal cell fate determination, cell migration, homeostasis, and growth. It also makes the cell susceptible to alterations that can lead to abnormal cell behavior and growth. So the E-cadherin cytosolic ends of the 5 repeats bind to the leaker protein, which is the B cadene. Then the alpha cadene binds to the beta cadene and forms a protein complex. Now when the adjacent cells are clustered together, the complexes then begin to assemble or remodel the adherent junction.
Okay, next we will talk about integrins. Integrins are proteins that bind to the extracellular matrix. They're heterodimeric and they contain alpha and beta subunit. Normally the alpha contain 18 subunits and the beta can contain 8 subunits, and they are non-covalently bonded. They transmit signals from the outside of the cell into the inside, and key components include tallin and kindlin, which bind to cytoplasmic tails of the integrins. The tails of the integrins are interacting with proteins in the cytosol, which eventually carry signals up. Once they're bonded, they trigger chemical pathways such as FAK and ILK. Okay, next I'll talk about focal adhesion, which basically connects the extracellular matrix to intracellular cells. Their cluster integrins interact with actin microfilaments and eventually go to linker proteins. And you have 2 linker proteins, which are the tallin and the A-actin. Tallin binds to actin binding proteins, and then A-actin binds to directly actin microfilaments. As stated, subunits bind to signal transducers such as FAK and integrin link kinase, which is ILK. With FLK, they are growth promoting, and they activate by autophosphorylation at tyrosine 397, and then it activates downstream proteins involved in cell migration. As far as ILK, there are scaffolding proteins that interact with domains of the B subunit, and they're involved with downregulation of E-cadherin expression.
Alright, so the crosstalk between E-cadherin and integrin-mediated adhesions. They have physical interactions between integrin and E-cadherin-mediated adhesions. Those interactions are MLC kinase and Rho-GTPase.
The MLC kinase, it's short for myosin light chain kinase. It belongs to the immunoglobulin superfamily, and the function of that is to phosphorylate regulatory myosin light chains and of the myosin 2, so it can facilitate myosin binding to actin, and that aids in the contractility by relaxing your muscles. The second is Rho-GTPase, and Rho-GTPase is part of is a small signaling G protein, and they're part of the RAS superfamily, and they regulate many aspects of the intracellular actin dynamics. And They are also found in the eukaryotic kingdom.
They're important for regulating crosstalk, controlling actomyosin contractility, and adhesion strength.
This has an impact on collective migration of the cells. When fibronectin, which are glycoproteins of the ECM that binds the integrins that are located on the substrate that are surrounded by E-cadherin molecules, they cannot form both integrin ECMs and E-cadherin mediated adhesions. When the amount of cadherin present is altered, the response of the cell ECM and E-cadherin mediated cell-cell adhesion changes the cell motility. It regulates the distribution of the focal adhesions and can control the directionality of the migration, but not the directionality of the speed.
CR and the FAC regulate both adhesion junction assembly and disassembly.
Cancer is basically an abnormal cell growth with the invasion of organelles in the body. There are two types of cancers, benign and malignant. Benign is basically where the cells grow only locally and cannot invade with neighboring tissues. They are often removed and usually do not come back, versus malignant cells where they can invade neighboring tissue and spread through body via fluids. They also can move away from other organs of the body and they can form other tumors. And there are over 100 types of cancers. This figure shows basically the difference between a normal skin cell growth and a tumor growth. And in normal cells, the cell division in the basal layer produces new cells that migrates upward and basically change the shape without dividing, versus tumor growth where the process is disrupted with cells that migrate upward and then divide, and that could be shown with the lighter cells and how you can see they're dividing and changing shape. The bottom pic shows the fate of dividing cells where the normal cells, each cell in the layer gets a rise to at least one new cell, where the light cells are going to the darker cells and there are no net accumulation. In the tumor cells, the division is not balanced and you can see they're branching off to newer cells, and it leads to a progressive increase in the number of dividing cells. And initiation is based off DNA mutations and how normal cells are promoted to precancerous cells. Next is promotion, where promotion stimulates outer cells to divide and form tumors, and it's a gradual process that requires prolonged and repeated exposure to the promoting agent. After initiation and promotion is followed by tumor progression, which is basically the mutations and changes in the gene expression, and it shows enhanced growth, and it's made up of a combination of DNA mutations and epigenetic changes. And after tumor progression, there's a repeated cycle that creates a population of cells that gradually changes. Okay, spreading of cancer. Spreading of cancer can come through proliferation, but it also can spread through angiogenesis and invasion of metastasis. And angiogenesis is basically the growth of new blood cells, and it was originated from Judah Folkman, who said that tumors release signaling molecules that trigger angiogenesis in host cells, and it's required for tumor growth. And basically, this is the fundamental step for transition from benign to malignant tumors. Once angiogenesis has triggered, then cells they can spread through two ways: invasion or metastasis. Invasion is the direct migration and penetration of cells into the neighboring cells, and metastasis is the ability for cancer cells to enter bloodstream and form tumors. Right here it shows the stages of metastasis, and there are 3 steps. The first step is cancer cells invade surrounding tissues and vessels, and basically in this step, they're different than benign, and they can leave initial cells and go to surrounding cells. Second step is the transportation. via circulatory system, where the cells penetrate walls of blood vessels and gain entry into the bloodstream. Lastly, it's the reinvasion and growth, where the cells in the bloodstream exit through the vessels and invade another organ and form metastasis that's far from the initial tumor.
Right. Now, knowing what cancer is and how it works, we can now look at it a little deeper and how it affects cell adhesion molecules and its function. Now we talk about E-cadherins as an invasion and tumor suppressor. E-cadherin is often lost in advanced tumors and have been linked to increased tumor reoccurrences. Loss of E-cadherins expression is usually due to the promoter of transcriptional repressors such as snail slug, CYP1, and ZEB1. This can cause epithelial mesenchymal transition, or EMT, which is the process by which epithelial cells depolarize into loosely organized migratory cells known as mesenchymal cells. This is a required process for embryogenesis, organ development, and wound repair. Being that mesenchymal cells are basically the basis for cancer cells, when they move from one part of the body to another, it's known as metastasis. When the circulating tumor cells exit the bloodstream, they undergo mesenchymal epithelial transition, or MET, to form new tumors at the sites that they end up. Okay, now we will talk about dominant negative mutations and how it affects E-cadherins. Dominant negatives are mutations that basically disrupts the normal effects of a wild type gene by overexpressing it. The mutation basically combines with the wild type gene, or dimerizes. In a polymeric molecule, it acts more like a deletion rather than a mutation, meaning that there's no production of that gene. In the experiment, we examined the function of cadherins in a normal cell. They introduced a dominant negative mutation of E-cadherin. It formed a complex with alpha, beta, and I believe gamma catenin. Cells expressing that dominant negative mutation resulted in a 5-fold decrease in the levels of endogenous cadherins and a 3-fold increase in the level of beta catenin. After all of this, the cell adhesion was inhibited. Next, we talk about the siRNA and how it affects integrins. siRNA basically stands for the small interferon RNA. It specifically targets a particular mRNA for degradation. It then interferes with the expression of specific genes with complementary nucleotides. In the experiment, they examined the crosstalk link, the crosstalk between integrins and growth factor receptor signaling, such as HER2. They transformed, I believe, the growth factor beta, which induced fat-dependent clustering of HER2 and integrins alpha 6, beta 1, and beta 4, and HER2 overexpressing mammary epithelial cells. It was mediated by ligand-induced EGFR activation and the phosphorylation of SRC and FAC. The knockdown of FAC, or the integrin alpha 6, by siRNA overrode the TGF beta induced receptor clustering and signaling to the PI3K and AKT signaling pathway. They also found that the inhibition of the SERC and FAC, inhibition of SERC and FAC reversed the TGF-induced resistance to the therapeutic HER inhibitor.
So, in conclusion, there are 4 cellular adhesion molecules. Those consist of the immunoglobulin superfamilies, the cadherins, the selectins, and integrins. And cadherins and integrins are vital to cell adhesion, and that's mainly because of how they interact with each other, and that interaction is called crosstalk. Cadherins, they hold 2 or more cells together by adherent junctions, and then integrins use their 2 heterodimers to hold the ECM to the hold their ECM to the cell. And if this process is ever disrupted, it could lead to tumor progression. Part of this is the EMT, or otherwise known as the epithelial mesenchymal transition. And next, there's the dominant negative mutation in siRNA interference. And that has been used in many studies to show how cadherins and integrins interactions are important. Also, there's a lot more work being done to discover the ability of inhibitors to stop tumor progression.